Hi, everyone. Welcome to A Moment of Science, Please. I'm Brian,
And I'm Mohammed.
and we are your hosts today for this episode. A Moment of Science, Please is a student-run podcast based at U of T, which highlights some of the fascinating scientific research we have going on here. Today, we'd like to welcome Dr. Navarre, Associate Professor at the University of Toronto's Department of Molecular Genetics. Welcome, Dr. Navarre.
Thank you. Pleasure to be here.
Yeah, of course. So to start off, could you please introduce yourself and sort of tell the audience a little more about you, as well as your background in education?
I've been a professor here at the University of Toronto for 15 years in the Department of Molecular Genetics. I'm a microbiologist by training. I got my doctorate from the University of California, Los Angeles, or UCLA, back in 1999. I did postdoctoral research briefly at NYU, and then moved labs to Seattle, Washington, where I worked for Professor Ferric Fong as a postdoctoral fellow in his lab. My work there, sorry, there's going to be a little bang, so let me turn off Outbeing and edit this out. I'm going to close my Outlook so I don't get notifications popping up. Let me see if I can turn off notifications. Well, anyways, let me just give me 1 second to recalibrate. Right. So I did a postdoc in the lab of Dr. Ferric Fong at the University of Washington in Seattle. And there I studied bacteria Salmonella, which most people know causes food poisoning. But I was particularly interested in the exact reasons why that is. Why is it that so many bacteria live in and on us and don't cause disease, but some bacteria do cause disease? And so I did some work there, and I figured out at the time that sort of how bacteria evolved to cause disease, some of the ways in which they pick up new genes to become antibiotic resistant, or to make a new toxin, for example. And that got me a position here at the University of Toronto. When I opened up my own lab, I was studying, and I continue to study Salmonella. But that's also gotten me sort of into some work on the bacteria that live in and on our bodies and in our intestines that don't get us sick as well. So that's where my research has sort of started to take a pivot recently.
So speaking of microbes that are in your body and don't, I guess, make you sick, I think you're talking about the microbiome, and I think a lot of your research is nowadays dedicated to the microbiota. So I was wondering what got you interested in this field and why do you think it's important?
Um, there's a lot of reasons I got interested in it. One was through a collaboration with another professor here at the University of Toronto, Dr. Alberto Martin, who's in the Department of Immunology. He had a mouse model of colorectal cancer, where if these mice lack certain DNA repair genes, they get colon cancer. And Alberto had an idea that perhaps it was microbes in the gut that were causing DNA damage to the cells in the intestine. And because these mice were unable to repair that damage, that they were getting increased mutation and therefore cancers. And I sort of consulted with him on that project, pointed him in the direction of trying different antibiotics. And we found that antibiotics did indeed reduce the tumor burden in the colon in these mice, suggesting that there was an immunological component. And then also consulted him on looking at the diet and its influence. And we found that certain diets would also change the susceptibility of these mice to cancer. And that this diet-dependent phenomenon required the microbes in the gut. So we sort of came upon this idea that diet impacts microbes, which then goes on and can impact things like colorectal cancer or colon cancer. What was interesting about that particular study is that the mice that Alberto was using actually represent the same mutations you find in about 20% of human colon cancers. And so I think this is actually a very relevant system to understand how microbes can affect things like cancer. These findings were published actually I don't know, 8 years ago. And they're not, a lot of other labs have come across similar conclusions. So this new interest in how our microbes both keep us healthy and how some of these microbes unexpectedly play roles in diseases like cancer or different neurological disorders, even autism, things like that, obesity, autoimmunity, all of these things, or protecting us from autoimmunity. All of these things, I think, have really sort of become apparent over the past 10 years, whereas we were completely ignoring our sort of gut microbes and other microbes that live in and on us as just being neutral bystanders. We don't think that anymore. And so it's an exciting area of research.
Yeah, it's really interesting stuff. So, because the microbiome consists of a lot of different bacterial species, do you often find yourself sort of focusing in on one species at a time, or do you like to study them as a whole? Or if there's benefits to both, I guess.
I think there's benefits to both approaches. One of the reasons why we haven't studied our own microbes and how they keep us healthy for a long time is because they're actually very hard to grow. Your intestines, let's say, contain about 300 different bacterial species, depending on what you define as a bacterial species. Some people say it's up to 1,000 bacterial species in your gut, mostly in your large intestine. Most of them have never been cultivated in a laboratory before. So the reason why we've had this explosion of knowledge isn't because we've started to grow a lot more of these bacteria in the lab, but it's actually because we've been able to do DNA sequencing and metagenomics is what we call it on tissue samples or fecal poop samples, and look at the microbes that are there without ever having to actually grow the microbes in the lab. And so we've gotten a lot of mileage, the field has gotten a lot of mileage out of this approach using metagenomics. But what quickly became apparent in our cancer study with Alberto Martin uh, was the fact that we didn't have the ability then to take individual microbes from the gut microbiota and put them back into mice and see which ones were affecting cancer and which ones were not. So we can do a lot of association studies looking at people who get cancer and people who don't and try to figure out through DNA correlations saying, oh, well, we see the DNA for this microbe in these people who get cancer. But there's plenty of people with the same microbes who don't get cancer, and maybe they will 10 years from now, who knows? And so we don't have the ability, without having the microbes in hand, to do the types of experiments that would be really informative to try to figure out how a given microbe impacts a disease like, as I said, obesity or autism or autoimmunity or cancer. And but the thing that slowed us down is that these microbes, they don't like oxygen. They're very what we call anaerobic. They won't tolerate even a minute of being exposed to oxygen. It turns out our intestines are very devoid of oxygen. There's no oxygen in your large intestine. And so a lot of people had a lot of difficulty growing a lot of these microbes. And so I saw that as an opportunity. And I think that it doesn't mean that the DNA sequencing work is less valid or not informative, but we do need a second approach as well of actually studying the bacteria themselves to learn even more about them and try to make better assumptions about what we see with the DNA evidence.
So we noticed that in a lot of your research, and as you've been explaining, that you use mouse as your model organism of choice. And you kind of mentioned that it's because of your collaboration that started out with that. But could you please expand more for our audience why mouse would be the best choice of a model organism for your studies? And also how would studying the mouse microbiome or the mice microbiome translate into studying the human's microbiome?
I think that's an excellent question. I think there's a lot of people out there who want to study the human microbiome, including myself. But the problem is that years and years and years, decades of prior studies on cancer, on obesity, on diabetes, on whatever, have been done in the laboratory mouse. And if we start studying human microbes and try to, let's say, put them into a laboratory mouse, we find that the human adapted microbes don't colonize or take hold in laboratory mice very well, if at all. So by having, and I'm not saying that we should not study human derived microbes. I think it's very important that we do, but we have, just a massive amount of data on laboratory mice with very little understanding of the microbes that inhabit those mice. And in the case of the Alberto Martin collaboration, when they were getting cancer, they weren't getting cancer with human microbes in their intestines. They were getting cancer with mouse microbes in their intestines. So one of the things that I would really like to do, and other labs are trying to do as well, a paper was recently published, is try to make a Rosetta Stone where we can translate what we see in a mouse better to what we see in the humans by saying, oh, if this bacteria X, I'm just calling it bacteria X, is associated with cancer in the mouse, is there a similar related bacteria in the human microbiome that might be doing the same thing to humans? But I think it might be a mistake to just take human microbes and just throw them into mice and expect the exact same thing to happen, or for any of these results like cancer or whatever in the mouse to suddenly replicate because you're using the human microbes. So I don't see my approach as being better. I see it as being parallel to other efforts made by other labs using other systems. And oftentimes where one approach is insufficient or fails to answer the question, there's some other approach, perhaps using individual microbes in a mouse, that actually ends up being more informative in that case. But really, I think we're only going to make progress in understanding how our microbiota affects health when we take a very holistic and multi-pronged approach towards understanding the relationships between us or any other animal and its microbes. And that'll include also, as I mentioned, looking at the diet. And, because the microbes that are in our body change depending on what we eat. And one of the things that we do, let's say even today, we can take a mouse that has no germs inside of it, colonize it with human microbes, then throw it back into a cage where we feed it mouse chow, not human food. And mouse chow is quite different than anything you or I would eat, but our gut microbes are used to seeing food coming down the intestinal pipe that is, a hamburger or pizza or whatever. And our microbes are not used to digesting or helping to digest mouse chow, whereas mouse microbes are used to, that's what they've evolved to do, is break down mouse chow. And so I think having an understanding of the fact that a laboratory mouse is not a perfect equivalent for a human, and also trying to figure out why it isn't and where are these differences, is going to be really important at improving our ability to take data we get from mice and then apply it to the human situation. Because right now, we have a mixed track record of taking data from mice and applying it to humans successfully.
Right. I think it's really interesting how much variation can result from like the different diets or conditions that an organism sort of experiences. Like I think Mohammad and I were actually joking the other day that like we've been eating a lot at the MedSci cafeteria, so our gut microbes are probably well adapted to that by now.
Yeah, I think that's something that people have to appreciate. I mean, you have to think about all the studies that have been done over the years. I mean, since I was a little kid saying, oh, if you eat this, you'll get cancer or heart disease. And this idea that our diet affects us directly, but a lot of our diet is processed by the microbes in our gut. And so I've become very interested in this concept of a diet, well, I call the diet microbe host axis, but it's a fancy way of basically saying how it's not just diet, and it's not just our microbes, but it's our diet being processed by specific microbes that can make a difference in how we respond to that diet. And so if I eat a slice of pizza and you eat a slice of pizza, a lot of the same metabolites will start showing up in our bloodstream an hour later as we absorb the breakdown products of that slice of pizza. But some of those metabolites will be different. And those metabolites that are different between you and me might be because you have different microbes in your gut than I do. And maybe if you have different microbes, maybe that will, make metabolites that make you more susceptible to heart disease or depression, or any number of conditions that we're starting to look at now. So I think looking at the microbes in the absence of thinking about the diet is short-sighted. I think we should look at both. And I think we should try to triangulate on how diet affects the microbiota and how metabolites produced by the microbiota from our diet affect us. So I think it's sort of it's a big question, obviously, and but I think it's going to start giving us a lot more aha moments when we start to understand, oh, that's why this diet is healthy for us and this diet isn't. And this is why this person, when they eat something, they put on 10 pounds and that other person stays skinny. I think that type of insight is waiting for us if we really look at the microbes as well.
So, in one of your recent publications on the collection of inflammation-associated mouse intestinal bacteria, you mentioned that you looked at the growth of isolates across different nutritional conditions. So, could you explain how you chose these conditions and what they might reveal about the murine gut microbiota?
Yeah, so this project was after we had done that work with Alberto Martin, the question became, okay, well, what microbes are causing colorectal cancer? And Alberto got a big team of us together, a number of researchers here at U of T, including Ken Kreuterud, Dana Philpott, Emma Allen Verco at Guelph. And my lab decided we would try to cultivate a whole bunch of mouse gut microbes and make a library, like a big collection of laboratory mouse derived gut microbes. The problem is, because they've never been grown before successfully in culture, we were trying to grow a bunch of bacteria that we didn't know how to grow. So it kind of became a circular argument. And our only solution to that was to just try as many conditions as possible. So we made up growth media of all sorts of nutrient compositions, and then just tried to isolate bacteria on each one of those growth media and then figure out what it was we had. Some bacterial species grew like weeds. They would grow on everything we fed them. Others would only grow on one type of media and nothing else. And we call those types of bacteria fastidious, but they're the bacteria that are very difficult to grow and have very particular nutrient requirements. And what's interesting about a lot of gut microbes, well, E. coli is a gut microbe. And I think it's actually a bad example of a gut microbe because it actually can grow on almost anything you feed it. E. coli is very easy to grow. It's probably why we do so much work with it. Other gut microbes are missing the ability to make all sorts of different metabolites. They can't make certain amino acids. Just like humans, we have essential amino acids that we need to eat to keep ourselves healthy. There's 9 essential amino acids for humans. Well, for many bacteria, they have essential amino acids as well. And they only get those amino acids from their diet, which is whatever we eat and makes it down the intestine to them. So what we learned from this is that the vast majority of gut microbes that we were able to cultivate when we looked at what they could eat or what they required for growth, a lot of them had very particular nutrient requirements. A lot of them were very stress sensitive. That is, that they would fail to grow if you hit them with a little bit of acid or a little bit of high salt concentration or something like that. So we were surprised, given how complex the gut microbiota is, how fragile a lot of the individuals in the gut microbiota, the individual bacterial species were, and how difficult and finicky they were to grow. What that tells you is that actually these gut microbes are used to being fed something by somebody else. Somebody else is providing the amino acids or whatever to them. And what we are seeing is a lot of cross-feeding relationships where symbioses form between different microbes, where one microbe will produce a metabolite that another microbe needs to eat. And so what you end up getting is actually a complex sort of ecosystem in the gut, like a forest, where you have like the alpha feeders that break down the food that comes in. They make secondary metabolites that then other bacteria come and use and turn them into something else. The whole time we're absorbing a lot of those metabolites across the walls of our intestine and into our bloodstream. And so they enter our bodily circulation that way. And so, yeah, I think I was shocked. The one thing I learned was just how fragile a lot of our gut microbes are. And this is now becoming a topic in the field because we're very concerned about the impact that antibiotics, that different drugs, that the Western diet has on the survival of our key gut microbes in our gut. And if we don't provide them with the right nutrients, as we change our diet to a highly processed Western diet, are we driving certain microbes extinct? And because our diet has changed radically over the past 75 years from being very plant heavy and lots of exposure to things like soil and stuff like that, to being very processed and very sterile. We do our best to eliminate microbes from food as much as possible. And so there's a concern that a lot of the diseases we now see in the Western culture diet, like multiple sclerosis or any asthma, allergies, lupus, things like that, might be exacerbated by the fact we're actually losing microbes that were highly prevalent 100 years ago before we started eating this way. So I think that's going to be a big area of insight and research.
Wait, so when does like an infant, in the embryo, I guess, when does the gut microbiome actually get established? Do they inherit it from the mother, kind of?
Yeah, so you're born pretty much sterile. And you first get exposed to microbes during birth as you pass through the vaginal canal. Breast milk itself actually has some microbes in it. But breast milk also has what we call breast milk oligosaccharides, or these chains of sugars. So it also contains lactose. So you, or a baby, when it drinks breast milk, gets a cocktail of lactose that it uses. The baby directly uses that. But then there's these other sugars that the baby doesn't have the enzymes to break down. And those oligosaccharides are actually there to feed microbes, to help a microbiota get established in the infant. So the mother is actually feeding not just the infant, but also the infant's microbes. The infant will typically, you have to realize, back in 200, 300 years ago, before we knew about germs, would be exposed to all sorts of germs that, just from handling and being carried around, and, mom didn't take a bath every day, et cetera, et cetera. So, the microbiota would get inherited from the people, mostly the mother and the family that was immediately in the surroundings. So you can't inherit a microbe that, is on a different continent from you. You only inherit it from people who are close to you or what you eat. So early stage microbiota is there basically fueled by mother's breast milk and what microbes like to eat those oligosaccharides from the mother's breast milk. And then when you switch to solid food, there's a massive change in the microbes that are present in your gut, and you slowly evolve the microbes that know how to eat your diet, I guess. And this isn't unique to humans. I mean, I think koala bears have a microbiota that's very good at breaking down eucalyptus leaves, and that would be the same for any other type of animal. Cows have a microbiota that's good at breaking down grass. And so, yeah, so I mean, the microbiota that you end up with is what you inherit from your relatives when you're young, and people around you, as well as some early ones that might come in on foods that you eat. And then by the time you're about 5 or 6 years old, you probably have a pretty complete ecosystem. And then it gets very hard for new microbes to sort of get a foothold in your gut microbiota, unless you take antibiotics or have some sort of a disease that wipes out your endogenous microbes or your normal microbes, and then you're susceptible to new microbes invading.
So I actually have a question. So if that's the way that the microbiome, I guess, develops in humans, does that mean siblings, or if not twins, have more closely related microbiomes to each other?
Yeah, that's absolutely the case. Is that people in the same family or people who cohabitate, especially when young, will have the same microbiota, or very, very similar. They'll be dominated by a lot of the exact same strains of the exact same species of bacteria. And you can imagine back, 500 years ago, when people lived in small villages and there was probably a lot of passing children around, you'd probably get the village microbiota or whatever that would be. So you'd see regionality that way. And we're a lot more, as I said, clean nowadays. Kids probably aren't as exposed not only to as many germs, but even to as many close contacts. And so, yeah, definitely, we expect, and what we observe is that, your microbiota is most similar to who you grew up with for the most part, unless you've had a major long-term treatment with antibiotics or something.
So one more question. Does that mean that the location you're in, I guess does geography play a role in your microbiome? So not just like the people you grew up with, because let's say you
Yeah,
move from one country to another, you're still with the same family but a different geography. Would your
yeah,
microbiome change?
definitely. But I don't know if I've never read a study that's done a good job yet at teasing apart geography per se. Like, are the microbes in the soil and the air of Canada different than in a country outside of Canada that somebody may have come from, or whether it's the changes due to lifestyle and diet. If you go from eating food in one country, and then you come here and you eat a lot of Tim Hortons and pizza, that will also have an impact, especially over a long period of time. I mean, if you eat if you go to a different country and you have one meal that's got weird food that you've never had before, your microbiota will probably shift temporarily a bit, where some microbes will bloom and others won't grow well. But it won't make a permanent change. But if you switch your diet for a very long time, it does make a permanent and lasting change in your microbiota composition. And it's all in response to the foods you eat, pretty much, and probably some of the microbes that you get exposed to.
So I guess as a researcher yourself, do you have any tips or advice for students who are interested in pursuing research? I know you teach a course about this, MGY280.
Yeah.
Brian
No,
and I
I mean,
both took different.
I'm just loaded with advice, but I think the most important thing is to, especially as an undergraduate, to try to get research experience and join a laboratory if you can. I think some students get very, especially young students, caught up in an idea that they want to study cancer, so they must go to a cancer lab. I would say that that's great if you can pull it off, but I think it's much more important that you get a good experience in a lab that will give you good attention and really teach you how molecular biology or cellular biology research is done, even if it's not, in this case, cancer, if it's, something else, but it's a really good lab. As opposed to, let's say, if you want to study cancer, but you go to a cancer lab and they don't have time for you and they ignore you and this and that, you'll probably learn a lot more and be a better researcher 10 years later by going to the lab that studied yeast, let's say, just because you learned how to think, how to mix chemicals, how to address a scientific problem, things like that. So if you can find a lab that really is willing to invest in you, I think that would be amazing. There's a lot of great labs at U of T. Finding positions is very competitive, and there's a lot of professors are very sort of, I don't know, I guess, their sleeve gets tugged on a lot, saying, "Hey, can you pay attention to me? And can you pay attention to me?" And so, it is challenging. But if you can find a really good research position as an undergrad, that's really key. And then I think for graduate school, then you can, when you apply for graduate programs, do your research, do your homework. There's all sorts of programs, not just in Canada, but in the United States and Europe and in Asia. That now you can narrow your interest more and say, now I really do want to do something with cancer. And then you have to do your homework and really look for laboratories that are funded pretty well and where the students seem happy and well mentored. Some labs look great on paper, but they may not actually be very good work environments. And some labs can be hidden gems. But I think the most important thing is to go someplace where you're supported, well mentored, and happy. And definitely at the undergraduate level, I wouldn't over focus on topic. Just kind of get topic adjacent. So cells and DNA, if you're interested in molecular biology, that's good enough for, if it's a really good lab, you'll learn something and it's translatable. So I think that would be my number one.
Yep, great. So by tradition, we always ask our guest speaker to tell us about their favorite scientist and why that is.
I've always gravitated towards scientists. There's a couple. One is a scientist at UC Davis named Andreas Boimler, who's in the field of salmonella, who for a long time was studying the less sexy. There was a lot of trends in microbiology where people got very excited about this thing or that thing, but he always started his questions sort of more focused on how salmonella can survive in a pig or a chicken or this or that. So it wasn't quite as sexy as what other people were studying, but you would read his papers and you go, wow, that's a really important question people should be asking. There's another guy named Harry Flint, who was early on in studying the gut microbiota and studied really boring stuff like how does the gut microbiota produce butyrate out of food? Butyrate is a short chain fatty acid. And he languished kind of in anonymity for more than a decade. And then people started to realize how important his work had been for the field. So I like the researchers who don't just gun for the sexy stuff, but they find an important question and work on it. And then other people start to slap their heads and go, oh, I get why this is so important. And then, sort of they're the ones who make it important, or they're the ones who make it sexy rather than following the herd. So these are researchers you don't necessarily hear about, because there's a lot of researchers who just dive into the next hot thing and just chase those things around. But I think the ones I admire the most include those two, and they picked an interesting problem and they stuck to it, and they did a really good job at hacking out some questions that probably from the beginning were hard to get funded, but eventually they both became really well established and really high powered labs.
So with that, Dr. Navarre, thank you so much for coming on today and sharing your research with our audience. It was a pleasure to talk to you.
Yeah, great. It was fun to be here.
So this concludes our podcast. Thank you to everyone for listening. If you found this podcast engaging, keep an eye for our next episodes. And don't forget to subscribe to us on YouTube and follow us on our social media.